So Matteo, over to you.
Hi, everybody. Thank you, Chris, for the nice introduction, and thank you for Twist for the invitation of doing this webinar. So, the title of the webinar is DNA Methylation Biomarker Discovery for Complex Diseases using Twist Human Methylome. And today on the webinar, we're going to talk about doing a small introduction on DNA methylation, what is biomarker discovery, what is the Human Methylome Panel, how at Diagenode we perform services using Human Methylome. Then I will present to you a short case study where we compare data from Human Methylome and from the EPIC array. And then I will explain how we can go a little bit further, going from a biomarker discovery to a biomarker validation, and then drawing some conclusion. But first of all, what is DNA methylation? So DNA methylation is an epigenetic mechanism that occurs by the addition of a methyl group to the DNA, mainly at cytosines. It can occur at different genomic locations, such as genome bodies, promoter, or non-coding region, like repetitive regions, small RNA, pericentromeric, or centromeric region. And the methylation, it is very well known that methylation can have an impact on gene regulation, mainly modifying the gene function or affecting the gene expression. It is also known that DNA methylation profiles are very specific to the cell type or to the tissue. And it is also very known that aberration of this DNA methylation profiles are an hallmark in cancer. And in cancer, we can find two main different mechanisms: a global loss of methylation, which is called hypomethylation, which can result in genomic instability, but also an increased DNA methylation, which is called hypermethylation, specifically at some region, mainly at CpG-rich region, which usually induce transcriptional repression. Knowing that, researchers know that now DNA methylation aberration of DNA methylation for cancer can be used as a biomarker for early disease detection. So how we can study DNA methylation? The gold standard to study DNA methylation is the use of bisulfite conversion. What is bisulfite conversion? Bisulfite conversion is a chemical reaction which transforms unmethylated cytosine to uracil, and then after PCR or library preparation to thymine, while the presence of a methylation over the cytosines will basically impede that modification to happen. And so after conversion, when we sequence the data and we know that on the genome of reference we have a C, and on our library we still have a C, that means that there was no conversion, so the C was methylated, while when the genome of reference we have a C, but we actually sequence a T, that means that the C was non-methylated and converted to a T. In the last over the last 2 years, we have also coming on the market with another option of bisulfite conversion, and a parallel option, which is the enzymatic conversion, which is shown here. And the principle is exactly the same, but instead of using a chemical reaction, we are using a series of enzymatic reactions to come at the same output. However, while the chemical reaction is very harsh in damaging the DNA, the enzymatic conversion is not damaging and will lead to a reduced loss of the DNA. So now that we have introduced the DNA methylation and why it is important to study DNA methylation, how can we use DNA methylation for a biomarker discovery, and what is the biomarker discovery? A biomarker discovery is a pipeline for which we can basically use a different sample that we know that we would like to find a sort of pattern that can differentiate one group to the other, to find a sort of signature. So for doing that, of course, we want to use the most genome-wide solution. So screen as much as possible CpGs to find the ones that are really important and can discriminate in between the different group, treatment versus control, healthy versus patient. And to do that, we can, of course, use different options. We have the option for whole genome bisulfite sequencing, or EMC, human methylome, the EPIC array. And those are very used screening high number of biomarker. And due to that, we should reduce and just try to avoid using a high number of samples. And here today in this webinar, we will focus on the biomarker discovery using the human methylome from Twist. But what is the human methylome? Of course, the human methylome is a targeted NGS-based technology that analyzes DNA methylation at single nucleotide resolution, achieving a very high coverage. So we are talking here about more than 150x. And just to compare, whole genome bisulfite sequencing is usually sequenced at 30x. So we have really a high difference here in coverage. How the Human Methylome Panel was designed? This panel was designed by Twist, and they have designed it using the recent database like UCSC, Ensemble, and ENCODE. And that panel basically covers more than 30,000 genes, which are connected to roughly 4 million CpG sites, and those 4 million CpG sites cover more than 80% of the CpG island in the human genome. And very interestingly, the Human Methylome Panel overlap for more than 90% the probes of the EPIC array. What are the applications for the human methylome? So the application, since it's very broad, it can be used for biomarker discovery and for monitoring in, for example, cancer, in neurodegenerative diseases, cardiovascular diseases, metabolic diseases, fertility, or immunology. And what is really nice about human methylome is that it's very flexible in terms of what are the sample type and the input that we can use for that technology. So we can go from really clinical low input samples, such as liquid biopsies and plasma cell-free DNA. We can use whole blood, PBMCs, flesh frozen tissue, but also FFPE tissue or cell culture in general. How it's important, so how we can use also the human methylome in cancer research. So if we cross the panel of the human methylome with a database, which is called the Network of Cancer Genes and Healthy Driver, we can find that, for example, the human methylome covers 277 genes that have been already reported to be important for breast cancer. It covers 150 genes that have been shown in this database to be important for colorectal cancer, 27 genes for the ovarian cancer, and 190 genes for the pancreatic cancer. So it seems to be a very wide panel that can be used for many different type of subtype of cancer. And together with Twist at Diagenode, we have been working since many years. And as soon as they have launched the human methylome, we started a collaboration together to be one of the first services provider to provide the human methylome services. And how the human methylome services workflow work at Diagenode. So we can start from a very different type of sample input, as told before. So it could be genomic DNA, FFPE DNA, cell-free DNA. We attach the adapters to those DNA. We perform the enzymatic conversion. And after that, we perform the library amplification. At that step, we have basically produced a methyl-seq genome-wide library preparation. So it covers the whole genome. And after that, there is the critical part. So we perform the capture using the human methylome panel probes, and we are basically just selecting for the part of the DNA which contains the important CpGs that have been selected by Twist in the human methylome panel. And after a small reamplification, we go for sequencing and bioinformatics. So the whole workflow at Diagenode have been optimized. And for example, if you submit a 96 sample, we can work in a full plate, and we can do that quite rapidly. So with a turnaround time about 6 to 8 weeks. And now I will present to you a case study where we have compared the data of the human methylome versus the EPIC array here at Diagenode. So a company, a US-based company called Inherent Bioscience, came to us with the idea of, with a pilot study to see whether it was possible, basically, to predict COVID hospitalization based on a DNA methylation profile of the patient, to see whether there is, we can discriminate prior whether there is a high or low risk of hospitalization. So for that, they collect blood from samples. They have selected PBMCs and extract genomic DNA. So we have got 4 samples, 4 genomic DNA from PBMCs for non-hospitalized COVID patient, and 4 samples from hospitalized COVID patient. And with the same DNA, we perform a pilot study with EPIC array and with the human methylome. So the first thing that we have done is to compare and see whether the methylation values on the probe of the EPIC array, which are also included into the human methylome, were giving the same DNA methylation values. So we have performed a correlation in between the DNA methylation values obtained with the 2 technologies. And here I'm showing to you all the 8 samples. And as you can see, we can see a very nice diagonal, and the Pearson correlation show a correlation of more than 95%, which strongly suggests that the two technologies are extremely valuable and interchangeable, even though the EPIC array is bisulfite based and RA based, while the human methylome is enzymatic based and NGS based. Then we have done a deep look at the EPIC array results, and we have performed a pairwise comparison in between hospitalized versus non-hospitalized patients. So in between the more than 700,000 CpGs that are analyzed by the EPIC after filtering NQC, we have found no significant DMCs, so differentially methylated Cs, and only 2 significant DMRs, which stands for differentially methylated region. On the other hand, looking at the human methylome results, what we have observed is that we have, with the human methylome, we're able to detect more than 18 million CpGs detected, and half of those, roughly 9 million CpGs, were covered with a high coverage. Of course, here we have to mention that thanks to NGS, when we say that the human methylome targets 4 million CpGs, but thanks to the Twist technology, which capture both strands, and to the NGS technology, which will sequence both strands to the plus and the minus, when we say that the human methylome targets 4 million, we are actually doubling that number, because every CpGs has a plus and a minus intuition. So we can actually also measure the methylation there. And that's why we have here 9 million covered with a high coverage. And when we have performed basically the comparison between hospitalized versus non-hospitalized on the human methylome results, among the 7.7 million CpGs that were common to all the samples, we have found more than 12,000 differentially methylated Cs and more than 800 differentially methylated region, when we have set the threshold at 25% methylation difference with a p-value adjusted of 0.01. And this, you can see it here in the volcano plot. And very interestingly, when we have used those more than 800 DMRs to perform a heat map and a hierarchical clustering, we have nicely observed that based on those 800 DMRs, using a hierarchical clustering, we're able to discriminate and separate very well between the 4 samples that were not hospitalized versus the 4 samples that were hospitalized. So those data, even though they are still a pilot project on a very limited number of samples, this strongly suggests that the human methylome, thanks to his broader coverage, is able to detect more easily DNA methylation signature that can occur in such peculiar or complex diseases. And after, so if we would have run that on a more higher number, just to be sure, not higher number of samples, just to be sure, and to finalize that those 800 DMRs and 12,000 DMCs are actually a very strong signature to discriminate between non-hospitalized and hospitalized data, then we will be able to move to a more custom target DNA methylation and moving from biomarker discovery to biomarker validation. And indeed, what is a biomarker validation? So the biomarker validation is the second step of the pipeline of biomarker discovery, where once we have finalized and find which one are the CpGs that are really important to discriminate in between the different group, healthy or treatment, cancer or healthy patient, then we're going to reduce the number of biomarkers only to the one that we are interested in, or the critical ones, and we can dramatically increase the number of samples just to validate the performance of the DNA methylation signature in a higher cohort, as it's shown here. And how this is practically done. So once we have performed the human methylome screening with the human methylome, together with Twist, we will work and perform a redesign of the Pro panel, going down, so reducing from the 4 million to the CpGs to the specific number of CpGs that have been shown to be very important. So in the case, for example, of the small pilot study that was done before, we were talking about only 12,000 CpGs. And together with Twist, we will redesign the panel, and then using a higher number of samples, we can go through the exact same workflow here as for the human methylome, just changing the part of the capture, and instead of using the human methylome panel, we can use the custom panel, which will capture only the region which contain the CpGs that have been shown to be important to discriminate for your specific biological question. And thanks to that, we can, of course, reduce the cost of the sequencing, because we're going to reduce the region that are captured and need to be sequenced, while increasing the coverage of those regions to be even more sensible for on those. So coming to the conclusion of that webinar, what I've showed to you today is that the human methylome is a targeted NGS-based DNA methylation assay, which is cost-effective to reach a high CpG coverage. So in reality, we have a more than 30x coverage of real targeted CpGs, and have a broader coverage than EpiCarry. Human methylome is, of course, ideal for biomarker discovery in cancer, but also in other complex diseases. We can apply the human methylome to very difficult clinical samples, such as plasma cfDNA or FFPE. And after the first biomarker discovery, of course, we can work with you together with Twist to reduce the panel from the human methylome and go into a more custom panel to perform the successive biomarker validation on a high sample volume, a high number of patients, to validate and to confirm that the signature is the right one to go through to screen many, many patients. And in conclusion, why do you want to use Diagenode as your targeted DNA methylation services? So we are happy to be selected by Twist as, or to be accredited by Twist as their NGS ProLab services provider. Of course, we have a huge experience in targeted DNA methylation project from difficult samples using many different panels, the human methylome, but also many other custom panels. We offer an end-to-end services from sample to the analysis. We always design the project in a very customized and collaborative way with our clients. So you will get you will have someone from our services team that will discuss with you the product design to design the best option ever. You will have a dedicated in-house expert for your project that will follow up on all the steps of the project. And we produce reliable and consistent bioinformatic data with high quality ready for publication. And with that, I'm concluding the webinar of today. I will thank you, all of you, for your attention, and I'm of course happy to take questions.